Hello, I'm Bobby Reiner, and I'm here to talk to you today about the life cycle of a modeling project. I'm going to specifically talk through investigating the potential impact of structured human movement on arbovirus transmission. So the goals of this talk are going to be charting the progress of a research project, thinking about the scientific process. We're going to be starting with the conception of the research project, the observations that drive the research project. We're going to move then into a hypothesis formulation. We're going to talk a little bit about the study design for this research project, how data was then collected using that study design, how we then tested our hypotheses, potentially refining, altering, or rejecting these hypotheses, and then finally followed through with the conclusion of the research project by writing and then publishing a paper. We're also going to discuss a little bit the development of a dynamic model that uses the data that we observe to develop new ideas for the study. And finally, as always, we're going to be stressing the central importance of data and the power of dynamic modeling. So the first step is really to make the observation that the world around us is complicated. What we need to do to have a fully formulated research idea is to distill our observations into a testable hypothesis. There are a lot of things we might observe that we might be able to try to explain in a way that, while being a plausible explanation, is too complicated for us to try to tackle in a single research project. And what we really need to do is identify a specific either observation or a specific process that is simple enough for us to try to actually test hypotheses around that observation, but not so simple as to be obvious. One of the key aspects of identifying testable hypotheses and looking at what our observations are telling us is going to be data. Understanding how the data that we might be looking at came about, understanding what kinds of processes the data might allow us to think harder about. These are going to be the issues that we're going to be wanting to think about as we move towards a simple enough research project that we can actually complete it, but a complicated enough one that will actually answer some interesting questions. The first thing we're going to want to do when we're thinking about the data is just look at the data. We don't have to start with a complicated model. We could just try to plot the data, look for patterns in the numbers, try to make tables, summary statistics. Are there averages that we can compare? Are there trends through time that we're going to want to look at and try to make additional observations from that? Finally, we're going to want to figure out, did those preliminary analyses, maybe just plotting the data, looking at it through time and doing a simple regression, that might actually be enough for us to assess our hypothesis. And frequently that might be enough to actually just produce a publishable paper, where we're looking at these straightforward statistical analyses. Sometimes these sort of straightforward statistical analyses are enough, but in many other circumstances, they're not really going to tell us the whole story, and they might fail to capture the dynamics of the system. One of the questions we're going to want to consider is, what kinds of data are going to be useful for modeling? If we're thinking about trying to create a dynamic system or a model world to understand some observation we've seen in the real world, how are we going to actually parameterize that? Or how are we going to inform that model world from the real world? What data might we want to use? This is clearly a critical question. Also, if we're trying to capture some dynamics from the real world, what are the known system processes governing the biological system? If we know, for example, we're dealing with a pathogen that has a direct transmission route, how are we going to incorporate that knowledge? If we think, as we're going to be going through in this example, we have a pathogen that is vector-borne, how much are we going to leverage our knowledge about the vector? How careful do we have to be? At the same time, what nuances of the system might we be able to ignore? What is vital to include? If we're trying to understand, for example, malaria transmission, how critical is it to understand the differences between the different mosquito species that might transmit malaria? Is it vital for our question? Or can we potentially ignore some of those species differences and still be able to ask and answer interesting questions? If we're trying to look at a pathogen that people have already studied before, there's probably various work that's been done on that. There's various models that have been fit, and from those there might be previously estimated parameters. How long is a person infectious? What's the probability of a severe outcome or the probability of death? How effective is a vaccine? These are all quantities that other people might have already estimated that we can use within our modeling efforts without having to worry about trying to estimate them ourselves. And then finally, it's always important, and hopefully I'll mention it a number of times, that science is a team sport. You're not expected to be the expert on the within human immunological response. You're not expected to be an expert on the actual implementation of the trial that collected the data for you. You're not expected to be the person who's going out and taking the blood samples, or perhaps the person who's running the PCR analysis. What's important is to be able to communicate with the different groups working on a project to identify where you can leverage their information to inform your models. Why even model at all? So frequently, we can collect data from a well-designed experiment and do basic statistics and identify the results we're interested in. In other circumstances, the problem can't be solved that way. We can't create an infinite number of universes where we can run a trial, sometimes for ethical reasons, sometimes for logistic reasons. And frequently, we're trying to learn something from the modeling that we couldn't learn otherwise. Additionally, sometimes the model itself isn't going to give us the final answer, but the model is going to suggest there are other aspects of the system we don't understand. And then, therefore, we should go collect new data on those specific aspects and try to further the work, try to understand how we're going to move towards finally understanding what's going on. And then finally, sometimes as we're building a model, trying to understand a world or an observation, the model itself will give us interesting results that we might not have actually expected when we started trying to answer our question. So we're going to talk through a particular example of this, but as is often the case, our story begins at the end of a different successful modeling project where we made some observations that seemed a little confusing and led us to go down the path that I'm going to describe. But first, I'm going to provide a little bit of background. Dengue virus is a mosquito-borne viral infection caused by any of 4 related but antigenically distinct virus serotypes, denoted dengue 1 through dengue 4. About half the world's population is at risk for dengue virus, and there's about 50 to 100 million reported cases of dengue fever, with, at this point, over 20,000 deaths from dengue hemorrhagic fever. There are a number of vectors for dengue, but the primary vector, Aedes aegypti, feeds throughout the day. So it's not just a dawn and dusk biter like some anophelines, and is also well adapted to urban and peri-urban environments. Critical for this analysis as well, later on we'll identify that it has a relatively short dispersal range, less than 100 meters. Frequently, an Aedes aegypti mosquito can live their entire life without even really leaving a particular home. But they do travel a little bit, but not nearly as much as other mosquitoes. The analysis that I'm going to be describing it focuses on some work that was done in Iquitos, Peru. So Iquitos is a city on the Amazon River, has a population of about 400,000. It's relatively isolated from other large cities. Dengue 1 showed up in 1991, Dengue 2 appeared in Iquitos in 1995, Dengue 3 showed up in 2001, and then Dengue 4 showed up between 2008 and 2009. So all 4 serotypes have shown up in Iquitos, but they came in a staggered manner. We also, part of the work that I was doing during my postdoc was with the Tom Scott lab at UC Davis. And they began doing intensive studies in dengue, of dengue in Iquitos in 1999, and were actually able to capture data of the invasion of both dengue 3 and dengue 4 as they moved throughout the city. I'm going to discuss one specific study that they were conducting. So in 2009, one of the things that they were trying to understand was the spatial and the spatial temporal spread of transmission. What they did was they set up a cluster trial. Starting with 2 specific areas of the city, they tried to identify how much movement was contributing to transmission versus how much mosquito movement was contributing to transmission. Within Iquitos, it's a about 15 by 5 kilometer sized city. Movement within the city is extremely easy. So there's no real restrictions on where people can go. We also had noticed by that time that transmission patterns weren't extremely local or focal. It wasn't that an outbreak would occur just in one part of a city. An outbreak would occur across multiple places within a city. In this analysis, before this analysis was conducted, human movement was hypothesized to be a driver of those spatial patterns. And so what was done was within 2 neighborhoods, active case detection was initiated to find actively infectious individuals. Once an actively infectious individual was identified, there then was an activity space survey done, trying to understand what homes they visited and then test every member of all of those homes. So it's kind of a snowball sampling to try to understand who else within their social community might have also been infected. For the purposes of trying to understand the differences between baseline transmission or background transmission and those that are actively infected, for each cluster that was started with a person who was identified as having dengue, another control cluster was also created. So for example, here we've plotted 2 of these clusters. What we're showing here are different houses that the person visited, as well as the number of people in those houses and whether they were exposed to dengue or not. So the way we read this on the left-hand panel, we have a dengue positive cluster. An individual was found. We're looking at the distances of all the other houses to that person. So there's their own home in the center of this kind of bullseye area. There are 4 people that lived in that home. And I can tell there are 4 people because I see 4 little spokes coming out from the center of my plot. 2 of those spokes are red. They look like a single red line, but in fact, it's 2 little red segments. That means that those 2 people were also actively infectious at the same time as this individual that started this cluster. We see 2 little blue spokes coming out. It looks like a single blue line, but it's 2 individuals telling us there were 2 other people in that house that weren't currently infectious, but became infectious in the next 2 weeks. We can also see there was 4 other homes that this person had visited. Let's see how well this works. We have 1 home here that was relatively close. We have another home here. These are each indicated by a number of spokes coming out from a centroid. We have another home over here, and then finally we have a 4th home up here. This 4th home, for example, has 5 individuals that live there. 2 of those individuals had appeared to have been exposed recently, but were no longer infectious. And then 3 of those individuals didn't seem to develop infection at any point in time during the trial or during the study. On the right-hand side, we see the same information for an individual that was just randomly selected. They had some sort of flu-like symptoms, but they didn't have dengue. We can see by counting the number of spokes, they had 2, 4, 6, 9 people living in their own home other than themselves. 1 of those people is yellow, had a recent infection. 3 of them became infected later on, and then 5 of them hadn't been infected throughout the entire duration of the trial. There were a number of other homes they visited, but one of the takeaways we have here is that there's fewer red spokes. There's fewer actively infectious people, although there was a lot of transmission that was going on at the time. So you see a lot of people who eventually became infected. We see this by looking at the blue lines that are coming out from the different houses. From this analysis, we thought, well, maybe we can build a statistical model to try to understand what's going on. We did exactly that. We tried to build a binomial regression model to account for the number of infections within each cluster home, looking at differences in district, seasonality, what year it was, and what the background infection rate was. Again, this trial took place in 2009, right as dengue 4 was showing up. So we had a lot of susceptible individuals, and then most of the people hadn't been exposed to dengue 4, so we had a pretty big outbreak. So we want to make sure we account for the background transmission rates. For the dengue plus clusters, we then added an extra term to say, is there some increased transmission due to the fact that we picked the individuals of this cluster and the homes of this cluster as a function of the first person we found being infectious? Lo and behold, we did find pretty substantial risks between the two different groups. The plot on the left shows the interannual variability. So there's different seasonality to when the transmission occurred. Red lines represent the expected probability of an infection for any individual in the contact network of one of these people that started one of these clusters. The red indicates the people who started it as a dengue positive cluster. The black indicates the individuals that were collected from a dengue negative cluster. So we still see substantial transmission, getting even in the baseline, the background control clusters, sometimes up to 40% of the individuals are expected to be somewhat exposed, or we're going to find some infection in those homes. And there was a substantially different risk between the individuals who were friends with someone who was actively infectious and that person had visited their home versus someone who had it. And the risk was independent from the distance from the home. That was one of the key things that we saw, was it wasn't just that when someone has an infection, mosquitoes get infected, they spread that infection to the neighboring houses. It was also that once a person is infected, other people they know were also infected. The conclusion from that paper was fine-scale human movements underlie patterns of infection and result in pronounced temporal and spatial heterogeneity in dengue incidence, basically trying to say that one of the key drivers of the spatiotemporal patterns was human movement. As we were looking at that, we also were taking some analysis of, well, how many of the homes that we visit when we start a cluster with an individual who's actively infectious, how many of those homes have at least one other person who's actively infectious? And versus if we start a cluster with an individual who's just got the flu, for example, how many other homes that person visited have someone who's actively infectious with dengue? Here, the red bars, this is two different seasons, season 1 and season 2. The red bars indicate that in the first season, we saw around 47% of the other homes had someone who was actively infectious. And in the second season, that dropped down to about 35%. On average, around 40% of the other homes we would visit would have someone who was actively infectious at the same time as an individual who initiated a cluster. There was a lot of transmission. So even in the clusters that were initiated by a control individual, we'd see 15% of the homes would have somebody that was actively infectious, which actually is a really high number. And so this led us to really try to question what was really going on, because that, while sounding like a plausible, lots of transmission, but I find a person who's actively infectious, I should expect to find a lot more transmission. It's actually really high. These numbers are, when we're going to start thinking about it a little bit more carefully and try to blend both this observation with the epidemiology of dengue, we're going to see that this is actually really hard to understand. So we're going to have to take a step back and look at the fundamental dengue epidemiology. Now, this plot, dengue has 4 serotypes. Those serotypes interact with each other. What infection you get when matters severely. For dengue, your second infection is the one that's most likely to be severe. Your third and fourth infections are very unlikely to be severe. Lots of complicated nuance. This is not what I mean by the fundamental epidemiology of dengue. I want to go back straight to the simplest model that describes both a host and a vector transmitting the same pathogen to each other. So we start here with susceptible hosts. They are exposed by being bitten by, let me see if I can turn my laser pointer on. They are exposed by being bitten by an infectious mosquito. They then go through an incubation period where they have been exposed, but they are not currently infectious. They are not going to experience disease symptoms either. They're in this exposed category. They then move to the infectious category where they themselves are now infectious to mosquitoes. A mosquito bites them, a susceptible mosquito bites them. The mosquito then has to go through its own incubation period where it's exposed, but not actively infectious before it can then complete the loop and become infectious again. Mosquitoes are hypothesized to be infectious until they die. On the other hand, humans are then able to recover. And I'm just focusing on any individual serotype. Maybe 40 years later, their immunity wanes and they become susceptible again. But this is the basic structure of dengue transmission. There's another key critical part of the basics of dengue epidemiology that make that observation before of 40% of the houses I visited have at least one other person who's actively infectious suspicious. Not necessarily suspicious, but very curious. This has to do with the timing of these infections and the duration of each of the time that people spend in these compartments. On average, once an individual has been exposed, it's around 4 days that they are kind of growing the virus internally, but they're not actively infectious. And this is indicated by this blue bar here from day 1 to day 5. Then they experience their infection. And they're also infectious for about 5 days. During that period of time when they're infectious, they might expose a mosquito to the pathogen. The mosquito then has an extremely long incubation period relative to how long the human is infectious. The mosquito's incubation period is around 10 days, sometimes a little bit longer. And from that point on, the mosquito is then infectious for the rest of their lives, probably. The human, who then might get bit by that infectious mosquito, is then going to go through its own incubation period before it is then actively infectious. One of the things that at least jumped out to us when we started thinking about this plot relative to those observations we saw from that study was there is a huge distance between the timing of an individual being actively infectious and the people that person might infect through a mosquito becoming actively infectious. If I'm infectious, I might infect the mosquitoes in my home. Those mosquitoes then might infect other people in my home. So it's not surprising that if I find an infection in a home, I might find another infection in the home later. What is surprising is to find other active infections. Now, taking a step back, it could be that I got bit by a mosquito at the same time as the other members of my home. That would explain this active infections in multiple people. But then the question is, what explains multiple infections at the same time in different homes? So again, this is the observation we had. We pick an individual who's actively infectious. We then try to figure out, okay, of the homes that they visited, how many of those homes have somebody else who's actively infectious? Now, again, based on the timings I was describing earlier, it's extremely unlikely that any of these people infected a mosquito that then infected any of these other people, because the time that would have to have gone by for these incubation periods would have meant that one of the people would no longer be infectious by the time the other people were infectious. So this is what we observed in the data. Around 40% of the other homes had at least one concurrently actively infectious individual. On the other hand, if we started a cluster and said, what houses did a person who didn't have dengue go to? We saw about 15%. Now, again, 15% is quite high, and this is going to explain some of what we see. There's just a lot of transmission going on. But this is the observation that we made. Okay, going back to kind of a little bit more generally, the development of model study concept. What's our question? So in our circumstance, our question is, what could have explained these patterns? What could have explained the fact that we have multiple houses that have simultaneous infections, and the way we found those houses was by asking a single individual, where have you been traveling? Why would this be interesting? So understanding what's driving the spatial patterns within a location, or what's driving the spatial patterns of transmission, will tell us something a lot more about what we might want to do in terms of reducing transmission, how we could detect more of the infections that might be going on, and will help us understand what is more important. Is it mosquito movement that's most important for understanding spatiotemporal transmission dynamics of dengue, or is it human movement? Who might be interested? Well, obviously, I would hope that dengue researchers are interested. Other vector-borne pathogen researchers might be interested, but we're starting to ask questions about human movement and how human movement contributes to transmission. Sure, in our circumstance, we're making these observations because of the fact that there's a mosquito involved, and that mosquito adds a delay or a lag between infections. But understanding how human movement contributes to transmission is something that hopefully people will be interested in for both vector-borne pathogens, but also directly borne pathogens, or directly transmitted pathogens. We're then going to ask a question as we're going down this process. How can we narrow down the question to a specific quantitative relationship. So I mentioned earlier, we've made an observation about the real world. The real world is complicated. How can we distill that observation into a question that can then be asked in one research project, as opposed to being the question that someone's going to spend the next 10 years of their life? Okay, so we then started asking questions about what hypotheses could explain what's going on. I'm just going to walk through what we don't think is happening. Okay, so if we have a person, and I'm going to color code things here, a house's color indicates whether there are mosquitoes in that house that are susceptible, have been exposed, or are infectious. People, and that's green, blue, or red. People are also going to be color coded the same way. A green person means the person's susceptible. A blue person means that person has been exposed, eventually will become infectious. And then a red person would be a person who is actively infectious. So here we could have a circumstance where we have someone walking around. They visited a number of homes. One of those homes has infectious mosquitoes. So we see they moved to the right sometimes, and they visited an individual who lives in that home. That home has infectious mosquitoes. That might lead to both of the person who lives at that home and my individual who's visiting 3 different homes, both being exposed. That's why both of those people are blue. They also visit other homes, but those other homes, the mosquitoes are not infectious, the people have not been exposed, so we have both green homes and green people. Once we then play this forward a little bit, once that person becomes actively infectious, they then are going to start exposing the mosquitoes. So they've been actively infectious, they've gone to the houses of their friends, mosquitoes have bit them, those mosquitoes have now incubated the pathogen. But then those mosquitoes would have to go through the period of that incubation before they become actively infectious. One of the things that this diagram would show is that we have actively infectious homes and actively infectious people at the same time. Which actually, while this might explain some of the patterns we've seen, this doesn't match the dengue, the epidemiology of dengue. It doesn't make sense that a person would still be infectious long enough for them to see homes they visited with actively infectious mosquitoes. There's this incubation period. As long as they're actively infectious, probably the mosquitoes that they've exposed are still going to be going through that incubation period and still expose themselves, but not actually infectious. On the other hand, if we wanted to try to understand why we would see multiple homes being actively infected at the same time, or having individuals that are actively infectious at the same time, we developed a hypothesis that maybe this was due to overlapping movement. Not only do I visit my friends and my other friends and my other friends, but all of my friends are friends of my other friends. And so they might all visit the same houses. And so while I visited a house that had mosquitoes that were infectious, so did other people I knew. An easier way maybe to think about this is maybe I have a family where I visit my uncle sometimes, I visit my grandmother sometimes, but my uncle also visits my grandmother. Because of that, we're all sharing the same risk at the same time. This would then explain a pattern where we see multiple people infectious at the same time. Put differently, if we were trying to understand how we think movement matters for transmission, we built the following kind of hypothesis about overlapping movement. We have an individual in the blue house. They have other friends that are in the same social group, maybe family, maybe friends. They visit those houses, but they also visit other houses. And of course, you're not expected to have the exact same friends as your parents. So maybe I visit some houses outside of my parents' social group. But in general, maybe there's going to be some overlap amongst these individuals. And then, of course, we're dealing with a mosquito-borne pathogen. Mosquitoes fly. So the red arrows indicate what we might see for the social spread, for the spread of the pathogen based on mosquito movement, not social movement. Distilled kind of into more of a conceptual diagram, if we think about the infection risk that we're bestowing on other individuals, if those individuals are families and friends, by visiting them while I'm actively infectious, I'm likely to expose mosquitoes in their home, which is then going to increase their risk of getting infected. Random individuals who I don't know, there's going to be some sort of background probability of infecting them that will probably be a function of how far away they are, because mosquitoes will will be contributing to that risk. But in general, if there's not much overlap in movement, it's unlikely for you to find me at their home, which means it's unlikely for you to find mosquitoes getting exposed due to my traveling around while I'm infectious. Okay, taking a step back to thinking a little bit more about how we could formulate this as a research project, we want to ask questions about who's tried to answer these sorts of questions before. What we're starting to talk about is network structure, network movement, and how people move around and how that contributes to transmission. Other people, of course, have done this before. Some people have done empirical studies where they've tracked individuals and found out where they're going, either using global positioning systems or surveys to understand what's going on, and to understand how movement and contact goes and contributes to transmission. There's been a lot of modeling studies on this. And so we're going to want to think about how have other people tried to answer these questions before we go off and basically recreate the wheel. Are there shortcomings in these studies? Are these studies exactly applicable to what we're trying to get at? Or is there something perhaps potentially that we can add and contribute to the study? And then finally, again, as we're going through, did some of these other analyses think about ways of quantifying the problem in useful ways? Have they used parameters or identified parameters that will be useful for ours? For example, when we're thinking about networks, we want to think about the connectivity of a network. There's language that's been developed about the connectivity of a network. There's particular statistics or quantities that people have developed and estimated. How can we use those in our own study? Okay, so we're going to start talking about constructing our modeling framework. What are our options? We've identified that we think that there's important movement aspects. We think how a person moves around is going to matter. Our hypothesis is that we have overlapping movement between individuals of the same social group. How are we going to incorporate this overlapping movement? How are we going to worry about things like maybe I have the same friends as my brother or sister, but my parents have a overlapping but different group of friends? Am I going to worry too much about mosquitoes? Do I have to go through the mosquito life cycle? Do I have to worry about pupae? Do I have to worry about mosquitoes laying eggs? Do I have to worry about weather? What are the aspects, the nuances I'm going to try to understand? Realistically, if I'm talking about people moving around, do I worry about mosquitoes? By the time I show up at a house, maybe mosquitoes have already conducted their blood meal of the day, or the blood meals they feel like they're going to take, and they might actually already be full. Do I have to worry about when I show up? If I'm going to start worrying about movement, what kinds of movement, how is it going to affect the rest of the system? What are the nuances I can ignore versus what are the nuances that I think are wildly important? Okay, within this, then we can describe what is the approach we're going to take. So I've been alluding to it. We're going to build a model where we're going to worry about human movement at an extremely fine scale. We're talking, the observation we made was about one house versus another house versus another house, where people were moving between these houses, and the movement of those people was driving the observation that we saw concurrent infections in multiple houses at the same time. Because of that, we're going to build a model that can track those things. We're not going to worry about the complexities of 4 serotype dengue. We're not going to worry about the complexities of importation. We're not going to worry about the complexities that are associated with a lot of the aspects of dengue transmission. We are going to worry specifically about the complexities associated with human movement at the household level. To that point, we'll build an agent-based model where we'll have a bunch of houses and a bunch of people, and those people will move around based on rules we give them. And then we'll be able to change those rules to see how overlapping movement matters. So therefore, we're going to build a giant simulation study where we're going to then have people move around. We're going to have the people get infected. We're going to have mosquitoes to some level, maybe not as nuanced as we possibly could. We're going to have this all happening at the same time. There's going to be a lot of stochasticity behind this, because otherwise it would be a very odd system. So we're going to have sometimes people get infected, sometimes people don't. Within an agent-based model, you're going to very rarely find a deterministic one. We're going to add stochasticity. Because we add stochasticity, we then have to simulate the model over and over and over again to try to get a better understanding of, did we not see an outbreak just because that was random chance? Because that person decided to move here versus there? Or did we not see an outbreak because there wasn't enough overlapping movement? So we're going to have to run our model many, many times and then ask questions about what we learned. As I mentioned, we're going to build an agent-based model of a neighborhood. We're going to follow this SEIR model for humans, where humans get exposed. They go through a period of time where they've been exposed. They then become infectious to mosquitoes. They then recover, but mosquitoes then go through the same pattern of from being susceptible, biting an infectious human being, to becoming exposed, then being infected, and then eventually the mosquitoes are going to die. So that's the modeling framework we're going to look at the individual level. And we're going to build a grid of houses, basically, again, looking at a neighborhood and say, how many people live in each house? We're going to give, on average, each individual, we're going to say each individual visits around 6 homes, which was about the average we saw in Iquitos. We're going to say each house has a member of a community of 6 different homes. So what we're saying there is, I have a group of people that are my community or my social group. Let's just say my family, for example, and that's made up of 6 homes. But the people I visit are going to be also 6 homes. Maybe it's perfectly those individuals of my family. Or maybe I don't really visit my cousin very much, but I do visit my mother, I do visit my grandmother, I do visit my uncle. So there's going to be overlap, but I'm going to be able to change the strength of that overlap. Specifically, we wanted to distill our question down into something quantifiable. We are talking about social structured movement. How much overlap do we see, and how strong is that overlap? How much do I have to move within my own individual group before I start seeing these patterns? As an example, if all that happens is I only visit my family and I never visit anybody else, and some other family only ever visits their family, we're essentially building islands across the city where people, sure, they live spread out across the city, but they don't visit anybody. In the absence of mosquito movement, this would result in very closed populations where we wouldn't see a big outbreak. So we probably don't want it to be perfectly overlapping. On the other hand, we're going to want to be able to quantify this in a way where we can say, how strong is our overlap of movement between members of the same social group or other social groups? And as we turn a knob, basically, do we start to match the patterns that we have seen? So what we're going to do here is we're going to define the level of within community movement by a single parameter. I had mentioned before, I visit 6 homes on average. I have 6 members of my social group or my family. And I could say, well, how many of the homes I visit are members of my family or my social group? And I'm going to govern that with a single parameter here. I'm going to call it kappa. So if kappa is set to 0, the orange homes are the homes that are members of my family. The homes with the black outline are the homes that I visit. In this circumstance, I visit none of the other homes of within my family. I only visit my friends, for example. So there's not much overlap between my movement and my social group. I could instead say maybe about half of the houses that I visit are within my social group. So now the houses I visit, again, are the ones outlined in black, but now 3 of them, other than my own, are orange. Maybe I visit my grandmother, my uncle, my cousin, but I don't visit my aunt. I don't visit my sister. I visit 2 other people instead. Or I could turn this knob and crank it, and I exclusively visit members of my own family. This is how I'm going to differentiate strength of movement. Finally, I'm dealing with dengue, which is a mosquito-borne pathogen. I have to care a little bit about mosquitoes and a little bit about mosquito movement. At least maybe I thought I did. And so I'm going to have a little bit about mosquito spread within the model. Here, I'm going to allow mosquitoes to move 1 house away. And so whatever the risk that I've seen in my house, it spreads a little bit to the house that's adjacent. Or maybe I'm going to see what happens if I allow houses to spread 2 houses away. This is going to be how I'm going to think about mosquito movement. Before I even get started with the actual analysis, we want to think a little bit about the limitations. I'm doing a pretty simplistic understanding of movement. Do I need to be more complicated? I don't know. I want to think about how much more I would gain by being more nuanced about what I'm doing. I don't have age in my model. I know that children are going to go to school. Adults are going to go to work. How is that going to change what's happening? Does it matter? Is it going to matter for me to understand anything about what I'm doing? Or have I simplified things so much that it's basically I'm not going to learn anything? I'm ignoring a lot of aspects of dengue transmission. I'm not worrying about disease. I'm not worried about detection. I'm imagining that I'm able to basically play God and know exactly what's happening within my system. I could try to add more of this reality, but am I adding reality in one way where I have this weird little grid of houses, and realistically, I probably should not add too much nuance in one place when I've already made so many simplifying assumptions in a different place? Finally, I also, as I'm going, I need to start thinking about, okay, what are those things I wanted to add to my system, but it's adding complexity. It's not necessarily clear it's going to help. Let's keep a list of those so I can make sure that I for future work, I can think about what I'd want to do. One of the things I might want to do is actually measure this overlap, this kappa. Is kappa a useful concept? This, how much my social group overlaps with the houses I actually visit. How strong is my social group? How many members are there in my social group? These are things I theoretically could measure. So then I get to writing my model. When you're writing a model, you want to be very careful because the model can get out of hand very quickly. The model I'm building, while it's in some ways straightforward, in other ways it's very complicated. I'm going to be keeping track of all these different people in all these different houses through time, worrying about whether or not they've infected a house or not. As I'm going, I want to think about what are the graphical outputs I'm going to use to display the answers to my questions. What are the graphical outputs I'm going to use to check to make sure what I'm doing is making sense as I go? I'm going to get in there, I'm going to write code. This particular code had one particular bug that took me a month to find. I won't get into it, but our documentation could be a little better. That being said, as you're writing code, you're going to want to think about version control. How are you checking to make sure your code's actually doing what you think it does? One of the hardest things is, even though code works, it doesn't always actually do what you think it's doing. At this point, I'd recommend writing your methods. You know what you're going to be doing. You know the experiments you're going to be running. Write your methods up now, and it'll help you understand what's going on. Okay, from our simulation, we did our simulation studies. We ran it many, many, many different times. We said, okay, how much does mosquito movement matter? How strong are these social movement groups? And we got some results. And we went with a particular figure that's got a lot going on to explain our results. Here we have both the data that started our observations as well as our results. So the data that started our observations were when you start with an individual who's actively infectious, and you think about the houses that person visits, how many of those houses have at least one person who's also actively infectious? We saw that was around 40%. That's why there's an orange surface at 40%. That's what we're trying to match for when you start with an actively infectious person, how many other houses have an actively infectious person? We also know there's a lot of transmission in the system at the time. Too much transmission will make every house have an actively infectious person. Or not enough transmission will have none, will not allow transmission to really take off. So here, the blue surface represents what we observed when you start with a not actively infectious person. How many people become infectious, or how many other houses have an actively infectious person? And we saw that was around 15%. So the things we were trying to match from our observations were 40% actively infectious individuals at number of houses or fraction of houses with actively infectious individuals, starting with a dengue positive person, and 15%. We then run our simulation to try to see how can we change what we see to make our simulated world match our empirical observations. On the x-axis, we see a mosquito movement. If mosquitoes are dispersing a ton, you're basically going to see the same thing everywhere across the whole city. Any infection anywhere kind of spreads to everyone's infected everywhere. That's why the two surfaces, the blue and the red surface, which are the results of our simulations, are very close to each other. So we can't have mosquitoes moving around too much, which is good matching reality because these mosquitoes don't move around too much. In the y-axis, we have how strong the network, the clustering of the groups are, how much overlap there is between where I go and where other members of my family go. I mentioned before, kappa being 5 means I only visit other people in my social group. When that happens, I basically build little islands where I'm only visiting people where I'm around. And if mosquito movement is set to 0, then it won't be surprising that there's basically no transmission. I only can infect people I know, and I can't infect anybody else. That's why if you look at the front left surfaces are going either to a very high or a very low number. But I do see there's a little bit of range where there's some overlap. It's pretty high. 4 out of the 5 houses that I visit are the same houses that my friends and family also visit. It's not 100%, but I'm able to intersect that surface. The red simulations intersect the observed orange flat surface. At the same time, I get the same thing for the dengue negative groups. I can kind of summarize this by saying, where do these things overlap? Am I able to find parameter values that overlap reality? And lo and behold, I can, which is nice for my simulation study. I have to have a lot of overlapping movement. I have to have a small amount of mosquito movement, but in that circumstance, I can recreate what I've seen, which is nice because it tells me maybe there's some validity to this and maybe I should keep going with this. Okay, now I've got my results. I want to start thinking about how to write a paper. First thing I'm going to have to do is I've made a ton of assumptions. Which of those assumptions might be wrong? I don't really know what the important assumptions are, so I want to start thinking about sensitivity analyses. What if I allow different movement patterns depending on age? What if I allow mosquito population dynamics to matter? What if I think about how well I'm actually going to be able to detect infections? Which of the assumptions are most important? If they're important, try moving them around, try changing them a little and seeing how much my results change. Sure, I might get a slightly different outcome, but if in general my qualitative results are the same, I'm not really going to worry so much about it. Just hitting it now, hitting it every time. How do I know my code's actually doing what I think it is? Just once again, what kinds of plots could I make? What results would I expect to see? If I say there's no mosquito movement, and I say I only visit my family and friends, I better not see a large transmission outbreak if I start one with me, because only my family and friends can get infected in that circumstance. Can I think of ways to make sure that my code's doing what I think it is? And then finally, are there other formulations I could try out? Are there other ways people have thought about these problems that are important? Okay. I'm now going to start thinking about writing up the results, the discussion, the abstract, and the intro. I want to be very clear about what I've done, but I want to be very clear about what I didn't do. I don't want to try to hide anything. I've made a lot of simplifying assumptions. You're always going to make a lot of simplifying assumptions. Are they important? Do you want to be very careful to not overstate how important what you've done is as a function of how many simplifying assumptions you've made? Try to wear a reviewer hat. Try to think, if you were reading this paper or this work and you were trying to think about what might be wrong about it, it's very hard to know how well somebody coded a problem. You're relying a lot on the results that they have. Try to wear your reviewer hat. On the other hand, don't do that too much. I've definitely had papers that have taken me years and years to write because I kept on thinking, well, maybe the reviewer is going to think about this. Maybe the reviewer is going to ask that question. Maybe the reviewer is going to want to see this particular sensitivity analysis. 50, 60, 100 pages later, I've developed this huge supplementary information that nobody read. Took me 8 years to get the paper out, but at least I felt happy. And you don't want to do that. Realistically, you have to move on. Got to pick a journal. Don't aim unrealistically high, but certainly, you want to get your work read by as many people as possible. That means you want it to be in a journal where many people read. These are high impact journals. It's not just for the purposes of promotion or moving on in your career. It's also the better the impact factor of a journal, the more likely it is to get read. You want to think a little bit about how long you want to go through this process. How many journals are you willing to try? Maybe you try one really high impact journal, then a medium impact journal. Do you want to just kind of get it over with? At some point in time, you just need to get it out. You got to move on. This is definitely a lesson that I haven't learned, but if you're a better scientist than I, you can try to, at some point in time, just say, okay, this paper is going to get read by the people I want to read it, no matter where I publish it. I'm just going to move on to the next one. In picking your journal, you can start also thinking about where are all your citations coming from. A lot of my citations come from PLOS neglected tropical diseases. So I send a lot of my papers to PLOS neglected tropical diseases. If the scope or the statement of a journal matches with what you're trying to do, it might make sense to appeal to it. If it is very clear that the journal you're interested in is for medical doctors and the work you're doing is a mathematical model, you might have a hard time getting that paper in that journal. You want to think about what other articles have been in that journal, what the audience is, how technical it will be. There's certainly a number of studies that have said the more math you have in a paper, the fewer people read it. That being said, that frequently makes your paper more understandable to those people who maybe are going to get the most out of it. And then, of course, finally, you want to think a little bit about how long your paper is, how long your paper is allowed to be. If your paper is already at 5,000 words, so you're trying to get into a journal that only allows 1,200, you might want to think about a different journal because it's going to take you quite a long time. Okay, I'm going to go back to one other thing. We sent this paper to I think we just sent it to Epidemics. We got reviewer feedback asking, we'd force these constraints. This kappa was very large. Do the strong constraints we enforce to match the observed patterns change the population dynamics? Like, is it very obvious that once you've forced overlapping movement to be so high to match your patterns, did you completely ignore the fact that it would be really obvious that's what's going on? Again, if people are only overlapping, I would have seen that in my system, because I would have seen there's no transmission between groups, only within a group. So are there obvious features of the data you ignore in effort to match your one specific feature? This is not so dreaded, but this is the famous reviewers experiment. Reviewers are going to ask you questions. They're going to poke holes in what you're doing, and they are going to request sensitivity analyses. You do not have to do all of the sensitivity analyses they request. However, if what they're saying is critically important for the major claim of your paper, you're probably going to have a hard time arguing against doing that work. So what we did is we said, okay, we've run these simulations. Let's take a look at the simulations we've already run. Let's say, how much did what did the overall outbreak look like? So this is the first plot we got to. So if there's no mosquito movement, which is unrealistic, but there's no mosquito movement, and you crank overlapping movement up considerably, there's a very obvious signature that this has happened. This is this dark red lines you see. Here's the many simulations we ran, the 5th percentile, the median, and the 95th percentile. You see there's a very obvious low transmission that occurred. On the other hand, if you allow there to be a lot of movement between and amongst groups, you get this green line where we don't have so much overlapping, where we weren't able to match the empirical patterns we saw, and you get what you expect to see with a normal kind of trajectory of infections through a group. So here, when there's no mosquito movement, it's really obvious that the overlapping movement of humans drastically changes transmission. And so it's probably a little unrealistic that we would have missed that. We probably would have seen that, or maybe we would have seen that within our system. However, and this was luck to some extent, when you have any mosquito movement whatsoever, whether it's a little bit or an absolute ton, that underlying obvious pattern of population level change and transmission completely disappears. What this means is, and we of course have mosquito movement because we're dealing with a mosquito-borne pathogen. What this means is, in this circumstance, even though we've added all this secondary structure to our system, once you add a little bit of mosquito movement on top of that movement, you can't tell just by looking at population level statistics what's going on. You have to look kind of under the surface. So our conclusions for our paper, well, first of all, we confirmed that our results were consistent. We didn't prove our hypothesis, but we at least didn't disprove it. Our hypothesis is consistent with the observed patterns of transmission. But then importantly, and this came only from the reviewer experiments, which in this circumstance is great. Sometimes the reviewer experiments are a pain. Here, this secondary structure was actually hidden. So it's a heterogeneity you only will see, even if it's strongly heterogeneous, you only see it when you actually look specifically for it. And if you're missing it, you don't get it. And then this was kind of the final goal. Infrequent as it is, that bottom line came directly from this reviewer experiment, where originally we were just trying to say social structure shapes dengue transmission. It's actually more interesting, perhaps, to say that it does this despite the fact that mosquito dispersal hides this structure. All right. So I'm going to finally conclude with a couple thoughts. Thank you for your attention. The first thing is try to work with the best data you can find. The data, this data was amazing, super detailed, allowed us to really worry about what's driving the problem as opposed to what's driving the biases in the data. Sometimes you have to worry a lot more about incorporating the biases of the data. Looking and playing at the data, this wasn't obvious. We wrote a whole paper about the results of this contact tracing, and only at the end did it start to kind of tickle the back of our minds. Wait a minute, how could we have had these patterns? What explains what's really going on? And it took us a whole paper of playing with the data to get there. We started with some simple summaries to really understand what's going on. We then moved to statistical models, and we found we weren't able to get to where we needed to get. We then went to the dynamic models. I don't know that you should jump into agent-based models right away, but in our circumstance, that's where we got driven by trying to understand what's going on. The data drove the model. We were always trying to match the specifically empirically observed patterns. And that was what we were trying to get at. As much as possible, we also derived or drew other parameters directly from the location. We tried simple solutions. We started adding more and more nuance to mosquitoes, and eventually it became very obvious that we didn't really need that. It was making computational time unwieldy because, of course, agent-based models take a while to run. And so eventually we simplified mosquitoes considerably. You want to think about what you're seeing, think about what you're doing, think about the results. It would have been nice if we had caught the hidden heterogeneity before a reviewer asked us to look for it, but at least we got there eventually. The second to last bullet point I can't emphasize enough. Publishing a paper is a frustrating process. You cannot take it personally. Some reviewers are grumpy. Some reviewers are mean. You cannot take it personally. Far too many great scientists have been turned off of science due to our current publication process, and it is exceptionally frustrating when you try to get a paper out and a couple years later you finally get the agreement. And then finally, of course, is probably more important than anything else, you should try to enjoy each step of the process. Probably don't enjoy the reviewer feedback so much, but everything else you should try to enjoy. And even reviewer feedback, try to find the actual useful comment in the angry ranting of somebody else. Okay. Thank you very much for your attention.